Hello, everyone, and welcome to Nature.com webcasts. My name is Hannah Viernes. I'm part of the market development team at Mission Bio, and today I'll be moderating the webinar. We are happy to present Dr. Chuck Gawad from St. Jude Children's Research Hospital. Dr. Gawad is an assistant member in the departments of oncology and computational biology at St. Jude Children's Research Hospital. He is a practicing pediatric oncologist whose lab aims to use single-cell sequencing to create a new generation of cancer genomics diagnostic tests that will improve the outcomes of cancer patients. Today, Dr. Gawad will be presenting on his work in profiling clonal hematopoiesis in cancer survivors with targeted single-cell DNA sequencing. At the end of Dr. Gawad's presentation, we will host a live question and answer session. To ask a question, please just enter a question at Ask a Question, and feel free to submit that at any point during the webcast, and we will answer that at the end of Dr. Gawad's presentation. Dr. Gawad, we welcome you.
Yeah, so thank you for the introduction, Hannah. It's great to be here. I'm excited to kind of present the first data that we've produced on the Tapestri Platform, and also excited to share, this unique cohort where we're able to look at childhood cancer survivors with this, exciting new technology. So just to give you an overview of my talk, as Hannah mentioned, I am a pediatric oncologist, so everything I do kind of goes back to, or really the place where I started actually is with my patients. And as a clinical fellow, was in 2009 when I started my fellowship. Sorry. So it was in 2009 when I started my fellowship, and I have an animation here, but I guess I'll just talk through it. It's not showing up. Was when genomics really came online, and I was really excited about the possibility of being able to use genomics to be able to improve the outcomes of my patients. But what I quickly realized after working in the lab was that tumor heterogeneity is an enormous problem, and that the only way we're really going to get at that problem is through breaking cells down into cancer tissue down into their individual building blocks, which are single cells. And so I've been working in this space for a few years to try and address those questions. As I mentioned, I have been doing this for several years, and so when I started doing this about 7 years ago, people asked me frequently, why do you care about single cells? And so this is kind of the analogy that I've settled on and been telling people. In the 16 and 1700s, people were looking at gross tissue. And in this example here, you can see the drawing of a brain where through gross dissection, they could identify new features of that brain and learn a lot of things that we didn't know before. But it wasn't until the invention of the microscope that we could really go down to the cellular level. And with that invention, the technologies have continued to improve, and what you can see here is that we can now see all these details and all these features of the human brain that we had no idea even existed before. And so similarly, kind of extending the analogy, we do bulk sequencing now, and everyone's familiar with bulk samples. I have a bulk cerebellum on the left-hand side and a bone marrow sample as well. And typically you mash up the whole tissue, and then you put it in the sequencer, and what you end up with is a readout that's a mixture of all the different cell types that you had present in that original sample. And so again, it's very hard to kind of deconvolute if you have different cell types, what was in there and exactly what was going on within that sample. So similarly, what I was saying is groups like Mission Bio and people in the field are really trying to develop these microscopes to begin to study genomics at the cellular level, and that there's really two parts to this process. So one is building new systems, and so we're talking about droplet microfluidics today, and the other is building new chemistry. And so that's what my lab and others have been working on, and that I'll talk a little bit about today. And so the readouts you end up getting are completely different than what we got before. And so instead of a single sample with all these different cell types, you end up with many samples with all the different cell types mixed together. You end up with a single sample, and you can identify the individual cellular components within that sample. But this process is technically challenging, and so there are a number of things involved in the process. And so you have to dissociate the tissue, you have to isolate the cells, you have to amplify the genome or transcriptome, you then need to interrogate those products. There's some bioinformatics challenges, and so the field's really been in development for a few years now, and we kind of went over this in a review a couple years ago that you can find there in Nature Reviews Genetics. And so, as you're all probably familiar, most of the work today has been done with single-cell RNA-seq, and so my lab has also done some work on single-cell RNA-seq, and these are just some examples of papers that we've published in the last few months. But DNA sequencing has been much more challenging, and it's really been what I've been more passionate about. And so the reason why is that tumors, have differences in gene expression, but they can also have massive genomic heterogeneity. And so in this now kind of classic example where they took tissue from different locations in the same tumor, what they found and did exome sequencing, what they found was that there were distinct mutations in each of the different locations. And so what it showed, even though, again, it's still at a bulk level, is that there's much more heterogeneity there than we appreciated. And it's not to say that we haven't learned a great deal from bulk samples. And so I was involved in this study, a part of a target study that was published last year. And we have cataloged mutations present in bulk samples and learned a great deal about cancer. However, there are some important limitations. So with bulk sequencing, we can only detect mutations in at least 10 to 20% of cells. We have a very limited capacity to determine if two mutations are in the same cell, and almost all the sequencing has been done in the original diagnostic sample. And so this comes back to my original point, which is that with bulk sequencing, we really haven't moved the bar a lot clinically. And so papers like this are starting to be published where you can see really only, in 5 to 10% of patients do genomics really inform a better treatment strategy. And so the question is then, why do we think single-cell genomics will be better? And so there are a couple reasons, so a couple important advantages of single-cell sequencing. So one is that you're getting a digital readout of every cell, and so the variant, if it's heterozygous, should be in about 50% of reads if your amplification is even. And so your detection is no longer limited by your sequencing error rate. And so if you look at 1,000 cells, your sensitivity, is 1 in a few hundred. If you look at 1 million cells, your sensitivity is, 1 in several hundred thousand. In addition, we also get the co-occurrence information of variants in each cell. And so it's important to know if you see two rare mutations, if they're in the same clone, that's continuing to evolve and is closer to becoming malignant, or if it's in two different clones. And then I'll show you a little bit of data on how we can see that. And so kind of to introduce the topic of the patient cohort that we're studying, so here at St. Jude, we've been treating pediatric cancer for about 50 years, and so, or maybe about 60 years. And so we have a cohort of patients now that are older, and it's very well characterized that these patients have chronic health conditions. So if you look at the purple line, those are their siblings compared to patients treated for cancer. They have a much higher rate of chronic health conditions. And so radiation is an important cause, but another important cause are some of the chemotherapy agents that we give. And one of the classes of drugs that are most commonly used are alkylating agents, and they are direct mutagens. And so the alkylation causes mispairing, and you end up with lots of mutations within cells. And so that alkylation, or that mutagenesis that occurs in these children that grow into become adults has been associated with cardiac dysfunction as well, among a number of other chronic health conditions. And so the other thing I wanted to introduce is clonal hematopoiesis of indeterminate potentials, so people call it CHIP for short. And so in the little cartoon on the left, you have normal hematopoiesis, where you have a stem cell that can self-renew, or it can differentiate into a progenitor that can also self-renew, or differentiate into mature cells. And so what people have discovered are these mutations that are associated with myeloid leukemias can be found in a significant proportion of the older population, and that they cause the clonal outgrowth of a distinct population. And so this is thought to be kind of a pre-leukemic state, and it's not clear the risk that these patients have into later on developing leukemia. I think people are studying this. But one thing that has come out of these studies is that patients that do have this clonal hematopoiesis have a much higher risk of cardiovascular disease, so a similar risk to smoking or high cholesterol or other similar risk factors. And so kind of putting this all together, what we wanted to study and look at in this study was this concept that long-term cancer survivors that received a lot of alkylating agent are at increased risk for this clonal hematopoiesis, and the idea is that this may contribute to their increased risk for other chronic health conditions, including cardiovascular disease. And so to study this, we used the Mission Bio platform, and so we have 5 patients with age and sex matched controls. They were all older than 50. They all received greater than 25 grams per meter squared of alkylating agent. And again, we had age and sex matched controls. The other thing is they hadn't undergone bone marrow transplant, so the hematopoietic cells that received the chemotherapy were still there as they aged. And then we also wanted to incorporate some phenotypic information into this study, and so we separated the blood samples from these patients into lineage positive and lineage negative samples. And then we did technical replicates, which I'll show you I think is still valuable to do at this point. And then we looked for an enrichment in CHIP-associated mutations relative to controls. And so I just wanted to step back for a moment here and talk about kind of single-cell DNA sequencing more broadly. And so as we discussed in our review, there are basically 2 important considerations. And so the main factor that limits what kind of study you do is sequencing costs. And so sequencing costs can come either from the number of cells you do or the amount of the genome you look at. And so depending on the study you want to do or the question you want to ask, you may want to look at more cells in a smaller part of the genome or less cells in more of the genome. And so this figure hasn't been updated, but the real advantage of the Mission Bio platform is that axis, the x-axis, can really be extended out, and people can easily now start looking at tens of thousands of cells, whereas in this study I published as a postdoc, it took us quite a bit of work just to get to 800 cells. And so just a little bit about the platform. So I think the solution really provides us with the capacity to do single-cell DNA sequencing in a high-throughput manner very easily. And so essentially how it works is you put cells into the system with beads, and then you lyse your cells, and then you add your amplification mix by merging 2 droplets, and then it performs PCR You can see the picture of the droplets merging in the lower right. And then it does PCR, and we use the myeloid panel that Mission Bio sells, which had 330 amplicons. And so you get a readout of each of those 330 sites, essentially a digital readout of each of those 330 sites with a large number of cells. And so the other thing that I'll bring up, and I'll show you kind of some raw data to show you some of the challenges that we've had to work through, is you then need to analyze the data. And so there are a couple steps to this, and much of this is supported by Mission Bio, but in my lab we've kind of worked on our own analysis pipeline just because I like to kind of deeply understand the data. And so the first step is you really need to segregate reads based on the cell barcode, and so there'll be sequencing errors and other challenges with that. You then need to take your reads, align them to the genome, call variants, and then you need to filter those variants for artifacts that were introduced. And as I'll show you, the filtering needs to come at multiple levels, so at the assay level, the cell level, and the sample level. And then when you have high confidence variants, you can then start to determine how different variants are, or how different cells with different variants are related to each other. So as I mentioned, there's some initial data processing, and so we developed our own pipeline where we just error-corrected barcodes, allowing one mismatch. And then we required a barcode to have at least 25,000 reads to be considered a cell. And then the things that didn't match, we discarded. And then we just use GATK best practices to do, or we use BWA to align in GATK best practices to do the variant calling. So in this slide, I just want to show, again, this data, any single cell data is not perfect. And so there's some differences in the depth in which you're able to, or you basically capture more target sites if you sequence deeper, but it will saturate at a certain point. So you need to be aware that you may not be getting perfect information from every assay in every cell, and so that needs to be an important consideration when you consider analysis downstream. So we've created these plots to kind of show you what the raw data look like. And so these are what some good examples of germline variants look like. On the left side, what you see, each dot is a single cell. Each row is a location in the genome where there's a variant called. And then the allele frequency is listed on the bottom. So you can see for each of those variants, most of the cells have it. So the proportion of cells that are different colors is shown on the right-hand side. And so what you can see is that each of the cells, each of those variants, the cells have an allele frequency that's hovering around 50%. And so those are really high confident variants, heterozygous variants, and that because they're in most cells, almost all cells, we would consider those likely germline variants. Alternatively, I can show you an example of a somatic variant. So in the very top, you can see a very clear germline heterozygous variant. In the two rows below, I just put those examples in to see some of the challenges with the variant calling. So you can see in the second one down there are a few mismatches, and so it's unclear what's causing those mismatches, and so we're still working on filtering criteria to try and get rid of some of these things that we think that we're not 100% sure is true. For example, the bottom row shows a number of variants that are called at very low frequency, so we think those are likely not true. And so we basically used this raw data to develop filtering criteria to identify the variants that we thought were true. In the middle, where the red box is, you can see an example of what we think, what looks like a very high confidence true variant. The variant is in TET2. It's at a known pathological site. And you can see the cells are hovering around 50%. So we feel highly confident that that's a true variant, although we are doing secondary assays to confirm that. So as I mentioned, I just wanted to show you what the raw data looked like, and we took that data and developed filters to try and really get down to the high confidence variants. And so the first filter we developed was an assay filter, and so if less than 50% of cells have less than 10x coverage, then we removed those assays because we didn't want to have assays that didn't perform in some cells and then believe that we have a clone there that's different because it's missing that assay when in fact it's just that it wasn't covered. We also have a cell filter, so we first called all the germline what we thought were germline sites, high confidence germline sites, and then if it was detected more than 75% of cells, and so we use that to identify the cells that had good coverage, and so cells with less than 25% of the germline sites called we considered not to be true. And then we had a sample level filter, so we only kept variants that were in that range where it had a number of cells with an allele frequency greater than 45%, and at least 5 cells. And so that will get rid of these variants that have low allele frequency that we're still kind of investigating where we think they come from. And then we had a patient level filter. So I mentioned in the beginning that we had technical replicates, and so and we had 4 samples from each patient, and so if we didn't find it in at least 2, then we removed that variant. And then just to mention, there are a couple of global filters. So one is related to GATK. So if a variant was within 100 bases of another variant, then we removed it. That's a known artifact of GATK. And we remove variants that are present in most samples. And so the idea there being that those variants are false positives, although we did go back and review the list, but the idea is that there's some systematic error in the sequencing or alignment that will give us these false positives. And so when we looked at our patients, again, we had 5 patients and 5 controls. We found evidence for clonal hematopoiesis in 3 of our 5 patients in the cohort. We didn't find any evidence for clonal hematopoiesis in the 5 age and sex-matched controls. When we look at the number of mutations and what the mutations are, so some of the mutations are known to be oncogenic, and you can see the range of mutations per sample was from 1 to 3. And so apologize that this isn't transferring also. Not sure what's going on. All right, just skip ahead here. So this also didn't transfer, but essentially what I can show you is in the patient that had 2 variants, we see no overlap in the cells with the variant. So it looks like this patient has 2 independent clones, one with a TET2 mutation, one with a STAT3 mutation, that developed independently, and so has 2 kind of what could be considered pre-leukemic clones. Conversely, we have this other sample where you have this P53 intronic mutation, although it has been reported to be pathogenic also. It's near splicing sites. But you have this P53 mutation, and so it looks like that was acquired first, so it's in about half of the cells. And then within the P53 population, that a subset developed also a BRAF mutation. And so at the top you can see that the red and blue are together, and so it looks like this is now a clone that has both P53 and BRAF mutations, so it could be kind of closer to developing into AML or MDS. And so in the last kind of parts of the presentation, what I was going to present is this concept that we started in the beginning from a piece of malignant tissue broke it down into cells, sequence each of the single cells, and we can read out measurements from each of those cells. But what we're also starting to do and starting to think about is how do we take information from each of those cells and start to put it together to kind of build a bottom-up understanding of tissue. And so we're thinking about ways to do this in my lab, and I think people in the field in general are moving in this direction. And one way that they're doing this is by measuring multiple classes of molecules in the same cells. So if you can measure DNA and RNA, or DNA and protein, or DNA and localization, or genome structure, you're able to kind of stitch together more information about the cell type. And more specifically, I think what I'm interested in is trying to connect genotype and phenotype. And so I think, single-cell RNA-seq gives you a lot of information about the state of a cell, and cancer specifically, but other diseases as well, are the consequence of the interaction of a mutation with a cell state. And so if you can measure the cell state along with the genomic changes, I think you'll have a much deeper understanding of how diseases develop. And so the way we're doing this in this study is to look at the lineage and positive and lineage negative populations. So basically what we're asking is, are there mutations that are preferentially found in either lineage positive or lineage negative that could either be promoting differentiation or restricting differentiation? And so the lineage negative population are the more immature cells, more immature hematopoietic cells. And so in this case, you can see the TET2 mutation, there's really not much difference between lineage positive and lineage negative. Similarly, for the STAT3, there is more, but probably not statistically different between the two populations. Similarly, with BRAF, you can again see that there's a little bit less in that population, but not significantly different. But then we do start to see other examples. So if you look at P53, you can see an enrichment in the P53 mutation in the lineage positive population. So suggesting, well, I guess that's, sorry, I apologize, it's not an enormous difference, so it's only a 5% difference. But the one where we did see a big difference is in the B core mutation. And so you can see that about a third of the lineage negative cells have a B core mutation, whereas only 15% of the lineage positive do. And so what this suggests is that this mutation is somehow restricting differentiation. Obviously, this is a very preliminary result and would require a lot of follow-up work to make that claim, but I just wanted to put this in there to make the point that by combining genotype and phenotype information, you can really begin to understand the biology at a new level. And so just some conclusions I had for the clonal hematopoiesis study. I do think that high-throughput single-cell DNA sequencing can detect aberrant clonal populations within tissues, including clonal hematopoiesis. But this isn't restricted to the hematopoietic system, so this could be, used to look at early-stage breast cancer, early-stage prostate cancer, areas in which, whether or not you're sure something's malignant or not, and you want to measure whether or not there's clones that are evolving towards a more aggressive state. Again, the two take-home points that I wanted to make were that unlike bulk sequencing, the sensitivity scales within the number of cells that you look at. And so you're really, again, getting a digital readout of what's going on rather than kind of relying on one single measurement. This concept that identifying different mutations in the same cells, I think, is really important. And so, again, if you see the sequential acquisition of mutations in the same clone, that suggests that that is a more aggressive clone, or that it's further evolved and undergone multiple rounds of selection for different mutations. But as I've shown you in the data here, I would also say that the field is still pretty young. So a lot more work's needed to improve both wet and dry lab protocols. I think the Mission Bio, the Tapestri Platform, is really an advancement in terms of throughput and the number of cells you can look at. But I think what you'll see is the technologies will continue to improve. And then the last point was really combining single-cell DNA sequencing with other readouts like surface markers opens up new strategies for really understanding how cell state and genotype interact to lead to disease. And then the last part of the conclusions are that, really single-cell DNA sequencing is poised to transform our understanding of human disease. And so, we start from a single cell and we end up with roughly 30 trillion cells. We each acquire a coding mutation every 300 cell divisions. And so we all have mutations everywhere in our genome. And how do those contribute to disease, I think, is an area people haven't really explored deeply. And, I said this a few years ago about RNA sequencing, but I think the same is true for DNA sequencing. I think you've seen labs that convert to single-cell RNA sequencing rarely go back and do bulk RNA sequencing unless it's on a really well-defined population. I also think, once these technologies are really moving forward and easy to use, that almost all sequencing for cancer and other diseases is going to be single-cell DNA sequencing. The amount of information you'll get will just, and the content of that information will just be much more important than what you can get from bulk sequencing. I also think connecting genotype and phenotype will be much easier at the single-cell level, where you can see combinations of mutations causing a certain phenotype. And so, as I mentioned, there are numerous basic science and clinical applications for high-quality single-cell DNA sequencing. And again, this concept that once we really understand and can measure tissues at the cellular level, we can then begin to think about how do we take that information and use it to build an understanding of normal and malignant tissue. And so, most importantly, I wanted to kind of end where I started, which is, everything that I do is motivated by my patients, most things that I do. And, I still believe that genomics is going to transform oncology, and that with single-cell genomics we're going to make even more advancements, but, constantly thinking about how do we uncover things and learn things that we can bring back to better care for our patients. So with that, I'd just like to acknowledge the people that have done the work. So specifically, Siva and Rob from my lab generated most of this data. So Siva did most of the wet lab work. Rob did most of the dry lab work. This is a collaboration with Les Robeson, part of the St. Jude Life consortium. And also Veronica and John contribute technically. And just wanted to thank Dennis and Dave from Mission Bio for technical insight, and with that, would be happy to take questions.
Great. Thanks, Dr. Gawad, for your presentation. So now we'll go to answering some of the questions here. And again, just a reminder, if you have any questions, please enter them into the Ask Your Question here and press Submit. Okay, so one of the questions for you, Dr. Gawad, how long will these patients be followed in the study?
Yeah, so St. Jude has a cohort of patients that they come every year to see the long-term doctors at the long-term follow-up clinic. And so as long as they're living in the area and able to come to clinic, these patients come indefinitely.
Okay, great. And so for cancers with many mutations, how do you select which mutations to test for? Is it previous biological knowledge or initial profiling?
Yeah, so I think you use both. Previous profiling will tell you which mutations you're most likely to see. So it's kind of a quantitative approach, but the other is you want some qualitative information. So if you know a mutation is associated with a really bad prognosis, even if it's not that common, you might want to include it because it might provide valuable information.
Okay, great. And another question for you. Are pediatric leukemia lymphoma cancers genetically silent? It sounds like we know that they're not, but maybe you could expand on that.
Yeah, sure. So I think that's a misconception that's been kind of propagated. When people present these pan-cancer studies and they look at the number of mutations per megabase, leukemia tends to have less mutations than most other cancers. However, the issue is that's again all made with in a bulk sample, and so when a patient's diagnosed with leukemia, they have 10 to 100 billion cells. A pediatric patient has 10 to 100 billion cells, and so that again started from one cell, and so that's 10 to 100 billion cell divisions, and again they get a coding mutation every 300. So there's lots of diversity there. And I think the diversity in cancer in general, so I suspect probably adult cancers have even more mutations. That's why you're seeing more in the bulk samples. But there's lots and lots of mutations in pediatric, in all cancers, where you have the outgrowth of cells. And so we're just beginning to have these tools now to start to study that.
Okay, great. And then another one. So you spoke about lineage positive and lineage negative. Can you specify better what you mean by that?
Yeah, so these are just the common terms used in hematopoiesis research. So lineage positive is a group of antibodies that are specific for mature hematopoietic cells, and so it will pull down all the mature cells. Lineage negative cells don't express any of those markers, and so those are the more immature cells. So the stem and progenitor cells are within the lineage negative population, and the mature cells are within the lineage positive.
Great. Okay. Did these patients in your study also receive radiation therapy?
So some of them did, yes, and that's an important consideration because radiation is also highly mutagenic.
Mm-hmm. And then there's, I guess, maybe some level of expectation that perhaps there was an increase in specific mutations that were associated with the chemotherapy in these cases?
Yeah, so that's the assumption we have going in. We're trying to also measure the per cell mutation rate in these patients compared to controls. But the hypothesis is that all this alkylating agent gave them a number of mutations that they then have to live with for the rest of their life, and that predisposes them to clonal hematopoiesis.
Makes sense. So another question here for you, Dr. Gawad. So can the patients have a better outcome with the combination therapy derived from the single-cell seq data? Is that the sort of hypothesis there, being able to target then genes that are present in all clones, for example?
Yeah, so not for this study, but that is the longer-term vision, is that, and this is something we study in my lab. So, the kind of overall hypothesis that we have is that the combinations of mutations within a cell determine the clinical behavior of that cell, or the phenotype of that cell. And so if they have mutations that make them, resistant to certain drugs, which there are very good examples in childhood leukemia, and they get a mutation that makes them grow faster, then those are clones that we see at relapse. And so the idea is that, if you had a really sensitive method where you could go really deeply within a tumor and find these bad acting clones, you would be able to change treatment and see that, know that certain drugs aren't going to work for those clones.
Okay, great. So another question here. How high is the prevalence of CHIP in your patient cohort, and is the spectrum enriched for certain gene mutations that are compared to the spectrum found in healthy elderly people?
Yeah, so we don't know yet. So this was just a small study, just 5 patients and 5 controls. There is a larger study going on with St. Jude Life using bulk sequencing, but again, doesn't have the same sensitivity as a single-cell approach. But the short answer is we don't know yet.
Okay. So question around how does single cell analyses then benefit a patient at the end? I think you spoke to some of the ways here, but maybe we could restate.
So the short answer is not yet, but the longer answer is that I think in the future that will be the case. So again, the important thing is connecting genotype and phenotype. And right now, when I do bulk sequencing on my patients, I get a list of mutations that are detected in their tumor. But 95% of the time, I don't know what to do with it. It's not connected with any action. What I do think is that having combinations of mutations in a clone and seeing a clone expand, if we can do enough studies to connect that genotype information with a phenotype, so NT5C2 mutations are associated with resistance to mercaptopurine, and so if I see a clone expanding that has an NT5C2 mutation, I'm not going to try and treat it with mercaptopurine. And so that is directly connected with an action. And so I think single cells is going to enable us to start to build out that information that can then later be diagnostic tests that we can use to change how we treat our patients.
Okay, great. So for these patients, and maybe this was missed in the beginning, did a mutant clone pre-exist prior to treatment, emerge as the clone was driving towards CHIP, or did completely new clones emerge? And how do you define the presence of CHIP?
Yeah, so I think those are good questions. I think whether it was a pre-existing clone, we don't know. We don't have any genomics data from before. However, if I had to guess, just knowing the prevalence of CHIP increases with age, I would suspect that they develop later in life. And then what was the second part?
And the second part of that was just how do you define CHIP presence?
Yeah, so I think that's an open question. I think if you have a mutation, in DNMT3A or TET2 that's known to be pathogenic, I think that's pretty easy. But if you have these mutations that are, of unclear significance, that's a harder thing to determine. And so I think people that are in that field are still, really working out those definitions.
Okay, great. And so how did your lab go about isolating the single cells? Maybe you could repeat the sample part of it.
Yeah, so part of the reason, not the reason why, but one advantage of working with leukemias is that we don't have to dissociate them. And so if you have to dissociate tissue, different tissue types can be very challenging. The approach that we took was really straightforward. So we took a blood sample, we phi-called it, and then we took out the mononuclear cell fraction, and then we just used the max column to pull out the lineage positive population, and then the ones that didn't get pulled down we used as the lineage negative.
Okay. So could you then comment on how many cells you need to sequence on Tapestri to confidently call these rare CHIP events in these blood samples that you processed?
Yeah, so I think the short answer is we still don't know. I think we need to do more samples to be able to answer that question. The more cells you look at, the more rare populations you're going to find. And so I think that's the real advantage of the Tapestri platform is you can do lots and lots of cells. And so I think more studies need to be done to be able to get, to start answering those type of questions.
Mm-hmm. And how many cells did you process, Chuck?
So in total, about 50,000. We could have loaded more, but we isolated the lineage negative population, which is pretty rare in the blood. And so, we were just doing about 1,000 lineage negative cells per sample. Another approach you could take would just be to load a lot more cells. But that's the approach we took. So you could do, I think you can do 10,000 on a single run. but we were doing, you know, a couple thousand per run.
Great. And you spoke through your analysis tools that you developed. Are these publicly available?
Yeah, so all the tools we used, with the exception of the cell barcode, which, you know, I know Mission Bio has their own toolbox, and I think, you know, the cell barcode extraction part there's nothing unique about that, and so that's something that could, you know, be, you know, there's no reason to use a fancy tool. And then downstream, we just use GATK, so those are all open source software.
Great. And just to say, for Mission Bio, we also have our open source tools available, and we've got sort of packages and software tools to help on the variant calling, along with the visualization analysis. So I think you talked a little bit about this, but maybe you could restate here your plan to sort of use and leverage this kind of method clinically in clinical applications.
Yeah. So again, I think the power is the two things are the digital readout. You know, more cells, it means more sensitivity, and sensitivity in certain clinical scenarios is really important. And then the second part is just not numbers, it's qualitative information. So, you know, what mutations are there and what combinations of mutations are there. And again, some of those are going to be more clinically important than others. But I think we're still doing, you know, more biology studies now to really build that information content that's going to later turn into diagnostic tests.
Mm-hmm. Yeah, and I know you've been focused on sort of the pediatric cancers here, but do you see, are there particular cancers that you think benefit from single-cell DNA analysis more than others, or do you see single-cell DNA analysis as sort of a tool for all cancers?
No, I see it as a important tool for all cancers, and you know, you can imagine one day having a patient come in and doing 1 million cells and being able to identify the clones that are really, you know, really important clinically and that have information attached to them that can change how we treat them. And I think that concept is important to all cancer. And even outside of cancer, we're learning more and more that other diseases have a component that's driven by somatic mutations. And so, for example, there was a paper in Nature earlier this year where they looked at the brains of patients that had died of Alzheimer's, and they had found evidence for APP mutations that were acquired somatically in those patients. And so not in it's a typical gene that causes early onset Alzheimer's, and they were finding it somatic mutations, not germline mutations. So I think we're still at the beginning of what we're going to learn on about how mutations, you know, at the single cell level contribute to disease.
Great. So obviously you've been focused on single-cell DNA sequencing, but what, if any, do you see clinical applications for single-cell RNA-seq, and why, or what are they, or why not?
Yeah, so I think single-cell RNA sequencing, and this is my opinion, but through my experience, but you know, single-cell RNA sequencing has taught us an enormous amount, and has been really important in creating atlases and in an unbiased way identifying all the different cell types and cell states within tissue. But I haven't seen, and you know, if you just look back to RNA sequencing in general, you know, RNA sequencing is used clinically to identify fusions, which occur in the DNA. Gene expression signatures, 98% of them that were published have never been used clinically. But we do, because they're really not robustly attached to phenotype. But we do routinely order DNA tests that are associated with prognosis or are associated with, sensitivity to a certain drug. So looking for EGFR mutations and whether you have a resistance mutation. And so, I think DNA is much more important clinically right now, and I think that will remain true as single cell moves into the diagnostic realm.
Great. Thanks, Chuck. Well, we certainly appreciate you for this presentation. I want to thank you for doing our webcast today, and then I want to thank all of you as well for attending this webcast. And if you have any further questions on the Mission Biotechnology, please feel free to visit us at missionbio.com. Thanks again, and have a great day.